close examination of the human immune system from a designer's standpoint reveals that the human immune system has the potential to upgrade its defensive capability. It can adapt to eliminate infectious agents, as well as cancer cells, despite the intelligence of cancer cells. This system is comprised of three components which include the human immune cells, the large intestinal bacteria colony, and the small intestinal bacteria colony. It is important that all components are healthy and maintain optimal communication within the group, in order to maximize the potential of the system. In cell-mediated immune response, white blood cells that recognize and identify invaders, called macrophages, are the first responders to foreign agents. Macrophages first engulf and digest invading cells, in a process that identifies the nature of the threat. They process the ingested agent internally, and then produce its chemical identifying marker, or signature on its outer surface, or membrane. This special signature is called an antigen. Special T cells, called helper T cells, are then informed of the threat through docking with the antigen cell signaling system. T cells are then stimulated to multiply, and secrete a signaling chemical which beckons other cells, especially cytotoxic killer T cells, to join the fight against the invading agents. Killer T cells docks with the invading microbe, or mutant cell, to identify it. It does this by interfacing with the cell's antigen, or membrane marker. Stimulated by the chemicals secreted by helper T cells, killer cells multiply at a rapid rate, and attack the invading cells, with bursts of cytotoxic chemicals. They then move on to destroy other cells. These cells also have the ability to continually upgrade their defense techniques, through communication with the colon bacteria colony. Therefore, the potential exists to develop new defense techniques, that an emerging cancer is unaware of. We will observe this process shortly. The large intestine, or colon, is the last part of the digestive system. It extracts water, salt, potassium and some fat-soluble vitamins from solid wastes, before they are compacted and eliminated from the body. It is the site in which bacterial fermentation of unabsorbed material occurs. The colon contains hundreds of trillions of a mixed colony of bacteria. These bacteria live in a mucosal film on the inner surface of the colon. The Bifidobacteria strain represents a large portion of bacteria colonizing the colon. However, unlike some species, Bifidobacteria are considered beneficial, and as such, exert a range of beneficial health effects. Bifidobacteria cells have the distinct ability to generate new attack methods against biological threats within their environment. This feature is very evident in the ability of bacteria to develop resistance to new antibiotic agents. Often, when Bifidobacteria is exposed to a threat, resistant factors are developed. These factors are actually microscopic DNA molecules, called plasmids. When the bacteria identifies a procedure that defeats the threat, the information is stored within its plasmid. Copies of these plasmids are shared and transferred among the bacteria colony, resulting in expansion of its defense. The first part of the colon, shown here, is designed to inform the immune system of new threats to the body, and to determine a response. The cecum and the appendix, shown here, are designed to allow for a reduction in flow of fluids and fibrous materials, just emerging from digestion in the small intestines. The low flow area is created here, as the digestive fluids gradually advance up the ascending colon. These low flow characteristics allow for close and careful contact between the massive bacteria colony hosted inside the appendix, as seen here, and the particulates in the food we eat that flow into the appendix. The appendix is designed to constantly take samples of our digested food, for further analysis. Now, we will see how, and why. We take a small slice of the appendix, to more carefully consider the immunological functions of this organ. Here, we observe four major components of the appendix immune response system. These are a blood supply, Peyer's patches, lymphatic supply vessels, immunological bacteria, and finally, immunological cells and antigens, or cancer-presenting cells, which we will observe shortly. The mucosal layer, or top layer of the appendix, is bathed in blood cells, as we can see here. But this blood supply is also a carrier for suspicious organic material, or cancer cell extracts, which have been gathered from all over the body. These extracts are carried on the surface of the immune system cells, and are called antigens. Here we observe a dendritic cell, presenting antigens, or cancer cell extracts, to the bifidobacteria colony inside the appendix. Bifidobacteria then formulate a defensive response and encode the details of that response in DNA strands called plasmids. These DNA chemicals are then transmitted to other bifidobacteria, thus enabling the entire colony to replicate the same defensive maneuver. These plasmid bacteria now contain valuable information about destroying the cancer that was introduced to them. They are then taken below the mucosal layer of the appendix. They are taken through a thin barrier layer, attended by M cells, not shown here, and are subsequently presented to immune system cells that populate the Peyer's patch below. The Peyer's patch is a part of the lymphatic system, and appear as circular patches under microscopy. Here we observe the macrophage cell, phagocytosing, or absorbing the plasmid. Macrophages will then present proteins, taken from the plasmid, on its outer surface, so that other cells may easily identify it. It then makes its way through the lymphatic network, where filtration, and the immune system response, may be expanded. The human lymphatic system is a network of vessels that remove fluids absorbed from the bloodstream and organs. This fluid is called lymph, and also carries immunological cells, white blood cells, proteins from blood, as well as various bacterial dead cells and cancer cell extracts. The lymphatic system is networked around small bean-shaped sites, called lymph nodes. These nodes filter out dead cells, or foreign materials, and also serve as a repository for immune system cells. Here, we can observe the macrophage carrying the bacterial plasmids from the appendix through this lymphatic circuit to the lymph node. The presence of this antigen inside the lymph node will stimulate the immune system cells hosted inside, ensuring that the critical information held in the plasmid is propagated and profilated among the immune cells. This can cause an even larger number of immunological cells to engorge the lymph node, causing it to enlarge. This expanded immune response now makes its way up the thoracic duct, along the spinal column, until it re-enters the blood supply in the left and right chest cavities. This critical information can be further propagated and profilated among the immune cells, and is used now to mount a formidable attack against the cancerous process. This may be repeated many times to reduce any attempt by the cancer at resurgence. The small intestine contains billions of lactic acid-forming bacteria, which also play an important role in conjunction with human immune cells, in eradicating cancer. While the colon bacteria provide the intelligence for the human immune cells to fight the cancer, the small intestinal bacteria provide vital supplements required to boost the fighting response. This includes important B vitamins and other organic compounds that are manufactured by these bacterial cells and absorbed into the body. The manufacturing process by the small intestinal bacteria is regulated by the bile produced in the liver and is secreted in the upper part of the intestine. A common alteration observed during this process is the increased secretion of citrulline in the bile, which encourages folate production, which is a critical vitamin for human immune cell activity. The human body uses folate to synthesize and repair DNA. Low levels of folate can lead to impairment of these functions, and possible cancer development.